In the final days of October, airplanes carrying top March of Dimes leaders, including CEO Cindy Rahman and Chief Scientific Advisor Dr. Emery Selly, descended on Texas. Once on the ground, they drove to the coastal city of Galveston and then to Dallas, preparing for launch. But there was no rocket, countdown, or blast off. Instead, the executives were after a giant leap for pregnancy outcomes, the end of preventable preterm birth. And Texas held the key. With its large, diverse patient populations, world-class research universities, and high-volume delivery hospitals, Texas has the resources and expertise to tackle the complexities of preterm birth. So on October 29th and 30th, March of Dimes opened its newest research hub there, the Texas Collaborative Prematurity Research Center, a partnership between March of Dimes and the University of Texas Medical Branch in Galveston and UT Southwestern Medical Center in Dallas. Led jointly by UTMB's Dr. Ramkumar Menon and UT Southwestern's Dr. Catherine Spong, the collaborative will pursue distinct complementary research tracks. At UTMB, principal investigator Dr. Ramkumar Menon is using pregnancy on a chip technology to study the biology of preterm birth and to test drugs that might reverse it. At UT Southwestern, Dr. Catherine Spong is conducting clinical trials with diverse patient populations and examining how nutrition, socioeconomic factors, the placenta, and bacterial vaginosis affect pregnancy outcomes. I've interviewed both scientists separately, with each conversation the subject of a standalone episode, available now. This is the episode starring Dr. Menon from UTMB. Here it is. Dr. Ram Menon is professor and director of the Division of Basic and Translational Research in Obstetrics and Gynecology at the University of Texas Medical Branch at Galveston. He's also one of two principal investigators at the March of Dimes Texas Collaborative Prematurity Research Center. Ram, welcome to the show.
Thank you for having me, and it's truly an honor to participate in the podcast with you.
It's great to have you here, and congratulations on the PRC.
Thank you. We certainly appreciate UTMB, really appreciate the honor and the award.
And we're very excited to have you part of the PRC family and our network of scientists trying to crack preterm birth. You're going to bring us a little closer to that reality, which our chief scientific advisor says, we will accomplish in this lifetime. So, Ram, you've been studying preterm birth for over 30 years. Suffice to say, your entire career. What drew you to this work? Why pregnancy? Why birth? Why mothers and babies?
Yeah, interesting, because when I was a graduate student at Wright State University in Dayton, Ohio, a master's student, looking for a project in infectious disease and immunology, my chairman of the department, microbiology and immunology at Wright State, introduced me to a young physician scientist who was interested in fetal inflammatory response as a contributor of preterm labor pathways. He was a maternal fetal medicine specialist. And I started a project with him looking at inflammatory response produced by human fetal membranes. And I saw the passion in him. As a clinician, he is very much interested in reducing the rate of preterm birth. He wanted to understand the fundamental mechanisms behind what leads to preterm birth. And that passion sort of grew in me. Whatever he introduced me to, the field that he introduced me to, the education that I received from him, the mentorship and leadership that I learned from him really, truly grew in me. And I've been working in the field since to understand preterm birth and to reduce the rate of preterm birth.
All right, let's talk about POC. Again, this stands for pregnancy on a chip. I want to give a quick background here for anyone not familiar with pregnancy on a chip. Ram, you helped me understand that this is an in vitro cell culture system a system that grows cells essentially that looks like a small glass slide you can hold between your thumb and your index finger. And I've actually seen a picture of your fingers holding this pregnancy on a chip online. That this chip is embedded with 14 types of human cells from 5 fetal and maternal organs that engage and communicate with one another. So we've got a bunch of cells talking to each other. They belong to mom and baby. POC is big for 2 reasons. First, it allows scientists like you and your team to better understand preterm birth, right? To watch preterm birth as it develops. And second, it acts as a testing platform for drugs that may reverse it. Did I get that all right? Is that all correct?
You got that all right. Yeah. So pregnancy on a chip is a one of the organ on chip platform that simulates an organ using cells. So these cells are derived from humans, so it's a humanized system. And in pregnancy on a chip, we recreate the entire human pregnancy related organs, as you said, on a small platform that you can hold in between your fingers.
Yeah. And this pregnancy on a chip does not exist in other fields. Obviously, you are the only scientist, you and your team are the only scientists in reproductive biology or maternal fetal health that are doing pregnancy on a chip. So this is absolutely unique to your lab. But in other labs across the country, in other medical disciplines, this exists. We've got liver on a chip, heart on a chip, kidney on a chip, skin on a chip, and even human on a chip, which is kind of this holistic system that you're doing, similar to pregnancy on a chip. So, like I said, you're the only one doing this in your field. Does it feel like the stakes are very high to succeed here? You're certainly pioneering this.
Absolutely, and we are aware of it. I mean, and we didn't start with pregnancy on a chip. We started with very small organs on a chip. I mean, pregnancy-related organs on a chip, such as lower uterine segment, upper uterine segment, placenta, fetal membranes, fetal brain. So we developed all these small organs on chips, and based on the success that we had, the lessons that we learned, we realized that a complete model is required. That means you cannot learn each organs in silos to understand human pregnancy, but we have to learn them all together. When they interact, they produce the actual physiology or pathology or whatever that we wanted to study. So eventually, 10 years of hard work by my team led us to develop these pregnancy on a chip. And there are several other labs that are doing single organs on a chip, like placenta on a chip, membranes on a chip, cervix on a chip. But we realize that alone is not going to solve the problems of prematurity or fully understand the mechanisms of prematurity or even do preclinical trials. So we have to have a complete system. That's why we developed the pregnancy on a chip. So it is, when we started this as a highly ambitious project, but we have built our model. We have tested our model, and we are confident that we can use this model now to do future scientific experiments.
Yeah, I love that. You need the whole system, right?
Right.
We're not operating. You hear it all the time. Pregnancy is an interconnected system, especially between mother and child, right? You cannot look at the uterus alone or the mother alone. There's inputs coming from the baby and vice versa. And also, as you know, the placenta is its own thing. You've got mom, baby, and placenta. So you've got 3 players there. They're all talking to each other. And you want to study that conversation that they're having. Okay, so I just want to clear up 2 misconceptions potentially that could be out there in the field about pregnancy on a chip. I said that it was a small glass slide. You helped me understand that. Initially, I thought it was a chip. I thought it was a computer chip. I thought you put it in the computer. This is not a computer chip, right, Ram?
You're absolutely right. This is not a computer chip. Like I said, I mean, it's an organ on a chip platform. You're actually simulating an organ using the cells derived from that organ. So it is really a cell culture platform in a microfluidic way, interconnected.
And there's no baby being grown on this glass slide, right? You are not growing a fetus.
No. We're not growing a fetus on a glass slide. Instead, what we are using is, you can actually grow a fetal organ. If you have the right cells, say, for example, if you have fetal brain cells, we can grow we can create an organ on a chip using brain cells, fetal organ on a chip. We can create fetal lung on a chip. We can create fetal heart on a chip based on if we have access to these cells. We can do that. But pregnancy on a chip will not grow a fetus on a chip.
Now, it's fair to say that preterm birth research has suffered because of the difficulty of testing drugs on reliable models. You cannot test on pregnant women because you don't know if they are safe on pregnant women. Non-human primates, not exactly the same as humans, quite close, but probably expensive, time-consuming. So how could pregnancy on a chip reduce or replace animal testing in pregnancy research? And why would this be a very good thing?
Right. You're 100% right that the current approaches that we have or current models that we have, whether it's a cell culture model, 2D cell culture model, or transplant, explant experimental models, or transwell models, which are using multiple cell types, or animal models, mouse, rats, guinea pigs, sheep. None of them are true indicators or mimicking human pregnancy-related changes. So, and you're right as well about the non-human primates. They are the closest that we can get, but it's costly, getting them not easy, and it is time-consuming. And you have to have specialists to operate them as well. So it is not an easy thing. So now how pregnancy on a chip can replace animal models. So our goal is 3 Rs: reduce, refine, and replace animal models in the future using humanized organ on a chip platforms. So how pregnancy on a chip can do that? The pregnancy on a chip has, as you mentioned earlier in your introduction about POC, we are recreating the entire human pregnancy associated organs on chip with 14 different cell types. On top of that, we also have the amniotic fluid compartment. We also have one of the fetal organs that we can recreate on this chip. So you have all the way from vagina to, let's say, fetal brain with the amniotic fluid compartment. It's recreated. We have the lower uterine segment, we have the upper uterine segment, we have the placenta, we have the fetal membrane, we have the amniotic fluid. These are all interconnected, and they interact each other. These cells interact each other. So a disturbance that happens in one of the systems, you can actually test and determine and the impact of that disturbance. Let's say, for example, vaginal microbiome changes. How does that impact the placental function? We should be able to monitor that. I mean, in a traditional system, that's almost possible. It's not easy to do unless you do multiple different experiments. Here on this chip, you can actually recreate that vaginal microbiome dysbiosis and test how that impacts placental function, for example. I'm just giving you an example.
Yeah.
So we recreated this model, and our first goal was to see whether our pregnancy on a chip, all these cells growing together for several days, have some kind of mimicry to human pregnancy-related changes. So we tested that. So we grew all those cells, we created the cell lines, we grew them on a pregnancy on a chip platform. Like I said, these organs are interconnected. The cell-to-cell interaction can be recreated. The endocrine signaling, paracrine signaling, all this can be recreated on this chip. So when we finished our normal pregnancy modeling, we collected these cells with the cell culture media, and we did a plethora of tests, multiomics analysis, image analysis, you name it. We have done a lot of tests. So the testing doesn't mean that they are great. So we have to have some kind of validation that it is really mimicking human pregnancy, or at least some form of resemblance to human pregnancy. So we took all this data and we compared this data with existing human-derived data sets that are available in public databases, NIH databases. And we also did our own studies by collecting tissues from human pregnancies and doing similar type of tests. We also did tests in non-human primate, normal human primate pregnancies. We collected organs and tissues from those pregnancies. We did similar experimental approaches. If it is an omics analysis, we did the exact same thing in monkey tissues and human tissues. If it is an image analysis, we did the same thing. If it is an immune cell trafficking, we did the same analysis. So all three were compared and tested whether pregnancy on a chip mimics human pregnancy-derived organs or non-human primate-derived. What we were able to find is more than 90% correlation of data with most of the organ systems that we tested, and that gives us confidence that we are able to recreate human pregnancy related physiology on this pregnancy on a chip. So if this is successful in the future, we have to carry out many more experiments as we propose to the March of Dimes. And once we carry out all these experiments, we will validate this further. At that point, we can say that this is much better than correlation to non-human primate pregnancy. We don't really need to use non-human primate pregnancy anymore for certain specific experiments. Now, just keep in mind, pregnancy on a chip will not deliver any babies here. So if you really wanted to test the delivery of the fetus, you still have to go back to an animal models. But what can we do with pregnancy on a chip? We can reduce, we can refine, and we can replace many of the experiments that we are currently using animal models for using this pregnancy on a chip. We can potentially eliminate many of the animal model studies.
Yeah. Well, so you've said two things here that I understand, Ram. The first thing you've said is we've been saying we've created a system that mimics pregnancy. We needed to test whether that was true, whether our statements were true, whether our experiment, whether the thing we've built is truly the thing we want it to be. In science, that's called validation. You validated the system, 90% correlation with most data points. That's big. So congratulations there. And you've also said an interesting thing too, another thing, which is that the pregnancy on a chip cannot deliver the baby. So you cannot, even though if you see inflammation happening, even if you see a reversal of inflammation, you cannot see the pregnancy outcome there because there's no cluster of fetal cells being born out of POC. So of course we're going to still need animal models there, but a massive reduction is possible here and has the potential for a big reduction. So that is fantastic. Now you have, in everything that I've read about POC and in having conversations with you, you've mentioned this term microchannels. The cells from each compartment, each organ interact with each other through microchannels. What are these? Because when I think of them, I imagine them as tunnels that, cells from the vaginal compartment can open the door and swim through the tunnel and meet and interact with cells from the cervix or the placenta and vice versa. Is that what it's like, or can you explain?
You explained it very well in a layman's way, and this is absolutely right. Microchannels are interconnecting the different organ systems. So that means the primary purpose of organ on a chip is how you can model intercellular interactions. So if you're doing a 2D culture, you're just isolated cells and you're growing them in a petri dish. Here, what you're doing is, you have multiple different cell lines that you're growing on a chip in different chambers, and they are all interconnected in a way that they are interconnected in utero, in a pregnancy environment. So vaginal cells can interact with the ectocervical cells. Ectocervical cells can interact with the cervical stromal cells. The endocervix can interact with the decidua cells, and the decidua can interact with the myometrial cells. All these interactions are modeled through microchannels. So these are small, miniaturized, as you said, tunnels, where if the cells want to migrate, can migrate. If they wanted to form junctions, they can form junctions. If the immune cells want to traffic in response to a stimulus, they can traffic. Extracellular vesicles or exosomes, if they wanted to go from chamber to chamber or cell to cell, they can traffic through these microchannels, and we can actually monitor them, how they traffic between these different chambers. So for example, if you wanted to recreate an ascending microbial invasion, that is one of the major factors associated with preterm birth, you can add the microbe to the vaginal epithelial cells. These microbe you can actually track the ascension of this microbe to the fetal side through different layers of the intrauterine system and how it reaches the amniotic fluid. And during that propagation of that microbial agent, you can also study, are they causing inflammation on their way? Are they colonizing in specific different cells? Are the placental cells producing antimicrobial factors? Is fetal membrane responding to these microbial invasion and causing some kind of rupture-like phenotype? All this can be recreated using this chip.
Okay, and here you're talking essentially about putting the pregnancy on a chip, the glass slide, into a state of disease, which then will model preterm birth, right? And you're
Yes.
watching how does that impact the cells?
Yeah.
Do they all become inflamed? Do some of them mount a defense against the inflamed cells, etc.? You're watching, and this is exactly what you're going to do, right, Ram? This is
That's
in a
exactly
couple
what
of
we
in a couple of months, in 6 months or something. You're going to actually introduce the cells to, on the maternal side, not on the fetal side, because you want to see if they travel, to a bad agent, right? And tell us about this. It's called LPS.
So what we are doing with the Master Times is that we wanted to now recreate a preterm birth mechanistic model. I mean, and
Yes.
as I said, infection is one of the major factors associated
Yep.
with preterm birth. So we are not going to put the live microbe yet. Instead, we are going to put one of the bacterial antigens, the lipopolysaccharide or LPS, which is a commonly used lab reagent to mimic infectious etiology. So
So this
we
is a
will
bad guy. You're introducing them to a bad guy.
and we are introducing a bad guy to the normal growing or normal otherwise healthy pregnancy, what we call healthy pregnancy. Now, how does the LPS causes inflammatory response on the maternal side? How does the LPS propagate to the fetal side and cause fetal inflammatory response? Is the fetal inflammatory response going to come back to the mother and forcing her to undergo contraction or cervical changes? Those kind of things are the ones that we wanted to test next. So we will be doing so we created a healthy model. Now we wanted to create a disease model. The disease model, it's restricted to infection for now, but that's the model that we wanted to create first. Then we can go on and test other factors. So, for example, if somebody is a smoker, has a high incidence of premature birth, can we recreate a smoker's pathophysiology using pregnancy on a chip? It is possible. So those are the future line of research. But we wanted to create an infectious model first.
We've talked about two things. We've talked about studying the healthy model of pregnancy using pregnancy on a chip. We've talked about watching the progression of inflammation in the cells in a diseased model of pregnancy. And the third most very exciting part of the PRC is actually trying to reverse that bad state of preterm birth or inflammation into that healthy pregnancy state. And you're going to be doing this with something called drug repurposing. Tell us what this is and how your AI drug repurposing efforts are going to work.
What we did is we 3D printed the feto-maternal interface on using our 3D printing platform. So we recreated the maternal-fetal interface using 3D printed platform. And what we have done using the artificial intelligence software, that we screen the drug libraries, existing drug libraries, from thousands and thousands of compounds that are already FDA approved or in the pipeline for FDA approval. We put some parameters on how we can select certain drugs for trials during pregnancy. So the AI ML based approaches, we can put some conditions. We want this. We cannot have toxicity to the fetus. It has to cross the fetal-maternal barriers. It has to have some ADME, the absorption. There are some pharmacological properties, ADME properties, what they call, and they meet those criteria. So based on that systematic screening of drug libraries that are already available, we have screened and we have narrowed down to about 350 compounds that we can initially screen using this 3D printed platform. Now, the importance of the 3D printed platform is it is a high throughput platform. So we can test all those 300 compounds in one shot. Whereas in pregnancy on a chip, as you know, that you have to grow 15 to 14 different cell types, 7 or 8 different people working at the same time to load these cells on a chip. It's a very complex procedure. But this 3D printing approach is a high throughput approach. So we will screen all these 300 plus compounds using this 3D printed platform and select the best candidate drugs that pass a bunch of criteria that we have set.
Amazing. Yes, Ram, let me clarify that I understand what you've said. Are you saying that you are not actually going to test these drugs on the POC? You're instead going to test these drugs on a 3D platform of the placenta. Is that what you're saying?
Maternal fetal interface. It could be placenta maternal interface, or it could be fetal membrane residual interface. Both of them are gateways of drug transport to the fetus.
Okay. Okay. And was this created with the same type of uh how did you create this? This is a completely different system from POC?
Yeah, it's a totally different system. It is actually a microfluidic platform, but it's 3D printed. So it's only have two different cell types: one mother, one fetus. So this is for screening purpose only. It is
Mm-hmm.
not going to test many of the things. So based on the high throughput screening, we will select a few handful of candidate drugs. Let's say top 10 based on the screening. We have criteria how we select these drugs. It cannot have cytotoxicity. It has to have efficacy. It should not produce any kind of inflammation on the maternal side or fetal side. It should not cause impact the viability of cells. It should not cause metabolism of the cells. The drug kinetics can be measured. The drug metabolism can be measured. All this, once we meet all those parameters, we will select the top 10, and then we will come back to a pregnancy on a chip platform.
Ah, okay. So it is involved. It is involved in the validation. Okay, it's just
Eventually, it will come.
okay when the candidates are narrowed down enough to be a
Correct.
small enough number to test, because this you don't have a million, you don't have a million pregnancy on a chips, and it takes a
Right.
very long time to culture these cells and etc. So okay, so you've now got about 350 promising candidates. You're going to use the 3D model to see which ones are actually the best of those, probably narrow that 350 to a 10, and then test those 10 drugs on the POC and see which ones of those best reverses the disease state to the healthy state, and also doesn't have any bad consequences,
Right.
unintended
Right.
consequences.
Right. Right. So then those drugs will move forward, potentially will move forward towards a clinical trial or
With
FDA.
women,
Well, yeah, yeah.
with pregnant patients. And Ram, most scientists I ask hate this question, but when do you think that could happen?
I'm very optimistic and think that if we can convince the FDA that these platforms are truly a representation of humanized system, and they are reliable and reproducible, this can move fast. This particular approach can move very fast.
Now, Ram, also tell me about this app potentially that could exist. Like you said, you want to move quickly. You were very smart not to give me an actual time. You never gave me a number of years or anything like that, but you said move quickly. I'm going to guess 5 to 10 years. That's quick in science, right? But you didn't say that. I did. But what about this app? Tell me. So it looks like there's obviously things that you're going to learn from the POC, right? There's biomarkers that will become apparent. All sorts of new pathways will be illuminated. You're going to put all that data into essentially an OBGYN app that a doctor can access, put in some risk factors that he sees, and that app will spit out potentially a medication or a course of treatment. Tell me how that would work.
So we are going to generate multi-omics data and different other forms of data, imaging data, biomarker data, as you mentioned, so many of these data. And what we wanted to do is, can we translate all these data into a predictive algorithm? Can we combine this with actual human clinical data and educate a model, a machine learning system? And can we educate a system to say that if you have very minimal information from a patient, such as history of the patient, some small biomarkers that they can test during either vaginal fluid or plasma samples or even amniotic fluid samples, using that minimal information and the algorithm that we can create from the pregnancy on a chip based data, that is mechanistic data, can we use that in a clinical setting to predict that this patient is at high risk for preterm birth based on such and such pathway that may be initiated by looking at a single biomarker or a couple of biomarkers or clinical variables? That's what we want to get it to.
So this
So
is a risk
as you
prediction tool. It would be a risk prediction tool.
hopefully risk prediction tool, as well as we also wanted to see whether we can define management strategies. I mean, since
Mm.
because we will have a drug testing component with pregnancy on a chip, so we can actually feed
Right.
that information and educate the algorithm that, if this patient is having infection and they get such and such drugs that may benefit placental inflammation, for example, or it
And
will
if
reduce residual activation of immune cells. But the ultimate goal is to develop a something really small, a chip that will go into your computer finally, that you can tell, or your cell phone, eventually can predict high risk.
Amazing. Ram, final question for you. Uh, you know, we've been talking about this question throughout the interview, but I just want to put it to you very concisely. What could preterm birth research and the preterm birth rate look like in 10 to 20 years if this technology succeeds?
Do you want me to answer that question? So I'm very ambitious. I think that I will put myself out of job. That's my goal.
Oh my gosh, that's wow. Beautiful.
That's my goal. And can I get there?
Your goal is to be unemployed.
Exactly. By answering the question that you just raised.
That's beautiful, Ram. Wow, how inspiring and optimistic. I love that. Ram, thank you for your time today and for your work on this incredible pregnancy on a chip system, for your 30-year career in the field. And I hope, I want to talk to you again. I hope 34?
35 almost.
35. Your work is translational and transformative. The world is watching. I know that you don't want to be at your job anymore because you want to solve preterm birth, but I do hope to have you on the podcast again, Ram.
Thank you. Appreciate it. Happy to come back on to your podcast again with new data, new news.
That was Dr. Ramkumar Menon, who leads the March of Dimes Texas Collaborative Prematurity Research Center at the University of Texas Medical Branch at Galveston. He's one of two principal investigators at the Texas Collaborative PRC. The other is Dr. Catherine Spong, who leads the PRC at UT Southwestern Medical Center in Dallas. You can find her episode right next to this one. Thanks for listening.